aromatic ring is there, then light will come and it will be confused and it will remain and inside will be absorbed. So aromatic ring is present in purine and pyrimidine. That's why DNA absorb light. Yes, tryptophan, phenylalanine, tyrosine, aromatic, absorb light. And porphyrias or porphyrin ring. Porphyrin ring are having four pyrrole ring. Porphyrin ring is having how many ring? Four pyrrole ring. So mnemonic, four pyrrole. So porphyrin absorb light at around 400 nanometer. If a person is having porphyria and we took sample, in this sample if porphyrin is present and you pass light through it, this sample will absorb light at around 400 nanometer. So there would be sharp absorption band at 400 nanometer. This sharp absorption band is called Soret band. So porphyrin, 4 pyrrole ring, 400 nanometer, that is Soret band. Sometime image-based question is asked. Now we come to basic amino acid, and mnemonic for this is his basics are loose, his not yours. His for histidine, basic means basic amino acid, R for arginine, loose for lysine. Whenever you will find basic word, immediately you will think of amino group, and amino group join with H-ion to acquire positive charge. So these are basic amino acid, they have positive charge. We found that histidine is least basic. That's why it is having only one positive charge or smallest positive charge on histidine. Arginine is most basic, so maximum positive charge on arginine. Lysine, intermediate positive charge. So we represent that histidine is least basic and we represent it by one positive charge. How you will remember? That English grammar we have read is RM. If one person is there, we say is. If multiple persons are there, we say are. He is only one positive charge. There are so many positive charge on arginine. He is histidine. There are arginine, so many positive charge on arginine. When we do electrophoresis, we found that on electrophoresis, cathode is having negative charge, anode is having positive charge. Don't get confused by name. Anode is not anion. Anode is having positive charge, cathode is having negative charge. And question is asked, which amino acid will move fastest towards cathode on electrophoresis? Out of these, histidine least positive charge, arginine maximum positive charge, lysine intermediate. So arginine having maximum positive charge will move fastest towards negative charge cathode. So answer is B. Arginine will move fastest towards cathode. Now we remember that basic amino acid are basic need in our body. Means basic amino acid either essential or semi-essential. We found that lysine is essential while histidine and arginine are semi-essential. How you will remember? Basic amino acid are basic need in our body, just like roti, kapda or makan. Three things. These are three amino acid. Out of these three, yes, histidine and arginine are semi-essential. Lysine is essential. If you see the structure of side chain of histidine, you will find that histidine is having imidazole ring. Which ring? Imidazole ring. I pronounce it as midday-jole ring. Midday meal ring. So midday meal ring. And histidine, dine, midday meal. And we found that histidine has maximum buffering capacity at physiological pH due to this imidazole ring. So again repeat, this imidazole ring provide maximum buffering action at physiological pH or in blood. So mnemonic is, yes, buffering as buffet, dine, midday meal. Buffet, dine, midday meal, you will remember. Now, we found that when histidine is metabolized, it is converted to FIGLU. That is form aminoglutamate. This FIGLU is further metabolized to glutamate, and in this conversion, folic acid, active form of folic acid is tetrahydrofolate, is converted to form aminotetrahydrofolate. So normally histidine is metabolized to FIGLU. question is asked that fig glue is a metabolite of, that answer is yes, definitely histidine. We found that if a patient is having folic acid deficiency, if folic acid is deficient, then fig glue will not be converted to glutamate and fig glue will be excreted. This 4-aminoglutamate will be excreted in urine. So in folic acid deficiency, fig glue will be excreted in urine. Fig glue test positive. So fig glue test is done for folic acid deficiency. Folic acid deficiency. So in the same question, if they ask that fig glue test is done for, then the answer would become A, folic acid. Other name is histidine load test. If you give loading dose of histidine, this histidine will be converted to fig glue. In folic acid deficiency, if you give loading dose of histidine, histidine will be converted to fig glue but will not be converted to glutamate. Fig glue will be excreted in urine. Fig glue test positive or histidine load test will be positive. So this is about fig glue test. Fig glue is a metabolite of. Now we come to arginine. Arginine is metabolized to NO. So it is precursor for nitric oxide. Nitric oxide do vasodilation. How you will remember that arginine, N-I-N-E, no. In Hindi, 9 is called no. And the name of enzyme is NO synthase. And this causes vasodilation and is responsible for erection. So if you are doing smoking inhibit this NO synthase and can lead to erectile dysfunction. All the movies, heroes are showing that I am smoking, I am a macho man. No, don't smoke. You will no longer be macho man in your bed. So don't smoke. Stop smoking. Yes, now you know the biochemical reason. Yes, NO synthase is inhibited by smoking. NO cause vasodilation, erection. So no erection, no macho man. So now next amino acid is acidic amino acid. Mnemonic is acid as glue. Acid as glue. S for aspartate, glue for glutamate. So aspartic acid or aspartate are same for question purpose. The only difference is that if you see this aspartic acid, inside chain there is acidic group. And you know, whenever acidic group is there, it will ionize to acquire negative charge. So aspartate is having negative charge. So aspartic acid or aspartate are same for question purpose, and you can say these acidic amino acid are having negative charge. In the same way, glutamic acid, yes, this is acidic group. It will ionize to acquire negative charge, glutamate, having negative charge. So acidic amino acid are synthesized in our body very easily. So they are non-essential. They are non-essential. And they are having negative charge. So this is important. A question is asked that which of the following amino acid is responsible for negative charge in fibrinopeptide A. I am not concerned about the structure of fibrinopeptide. I am concerned about the property of amino acid. I know that this answer may be aspartate or glutamate because you know aspartate and glutamate have negative charge. So answer was aspartate was given the option. So acidic amino acids are having negative charge. Now you can remember that aspartate is having 4 carbon, 1, 2, 3, 4. Glutamic acid is having 5 carbon, 1, 2, 3, 4, 5. You will remember alpha ketoglutarate, glutamate, glutamine, all are having 5 carbon. Again, I'm repeating alpha ketoglutarate, glutamate, glutamine, 5 carbon. Gluta means 5 carbon. Now, aspartate, how many carbon? 4 carbon. Now you can make a mnemonic. If you join 1 carbon with the help of glue, means aspartate 4 carbon, and I joined 1 more carbon with the help of glue, it became glutamate. So aspartate 4, yes, and glutamate 5 carbon are there. And you know, whenever amino group is there, ammonia is there, it can bind to carboxylic group. So if aspartic acid bind with ammonia, it will form asparagine. This is asparagine having this amide. group. So amide amino acid, asparagine is amino acid having amide group. Glutamate or glutamic acid bind with ammonia to form glutamine. So glutamine, that is having amino group. So this is about acidic amino acid having negative charge. These are amide. Amide amino acid are not having negative charge. They are polar. They are polar without charge. Polar without charge. No charge on asparagine or glutamate. Now essential amino acid we have discussed. Mnemonic is his, mat, will, fly. So histidine, arginine, they are semi-essential. And lysine, basic amino acid, that is essential. Aromatic amino acid, tryptophan, phenylalanine, essential. Branch chain amino acid, these three are essential. Will. Threonine, OH containing amino acid, essential. Sulfur containing amino acid, methionine, is essential. So mnemonic, simple mnemonic, yes, his, mat, M-A-T-T, will, V-I-L, fly, P-H-L-Y, fly. These are essential amino acid. Now, we can classify on the basis of polarity. Non-polar amino acid, you remember very easily, all aliphatic amino acid are non-polar except glycine. I told you, according to Harper, glycine is classified in polar amino acid without charge. Polar without charge. Now, aromatic amino acid, phenylalanine, tyrosine, tryptophan, all are non-polar. Amino acid proline is non-polar and sulfur containing methionine is non-polar. So you can easily remember these four groups. Now we have just discussed basic amino acid are basic need. They are essential. And they are having positive charge. Yes. Acidic amino acid are non-essential, are having negative charge. So they are opposite to, they are, yes, polar with charge. Other than these two groups, you can say all remaining amino acid are polar without charge. So now I can conclude that alpha ketoglutarate having 5 carbon join with ammonia to form glutamate. Glutamate bind with ammonia to form glutamate. In the same way, oxaloacetate having 4 carbon join with ammonia to form aspartate, and aspartate join with ammonia to form asparagine. So these are reversible reaction, you can say like this, transamination reaction, transfer of amino group occur. So I explain, this is a mnemonic type of thing. Now I explain transamination reaction, transfer of amino group. First is alanine. Alanine is having this amino group. Alanine transfer its amino group to alpha ketoglutarate. And whenever you transfer amino group to alpha ketoglutarate becomes glutamate. So we found that when amino group of alanine is transferred to alpha ketoglutarate, this alpha ketoglutarate is converted to glutamate. This is converted to glutamate. And when you remove amino group from alanine, it becomes pyruvate. Alanine becomes pyruvate. So this is transamination reaction, transfer of amino group, and transamination reaction cofactor is vitamin B6. Vitamin B6. And the name of enzyme is SGPT. S for serum, G for glutamate, P for pyruvate, T for transaminase. Another name is ALT, that is alanine, AL for alanine, T for transaminase. So in this way, alanine transfer its amino group to alpha ketoglutarate and it is converted to glutamate. I remember this. Another transamination reaction, aspartate, transfer its amino group to alpha ketoglutarate. When amino group is joined to alpha ketoglutarate, it becomes glutamate. And aspartate, when aspartate amino group is removed, it becomes oxaloacetate. I told you the only difference between aspartate and oxaloacetate is amino group. So if you remove amino group from aspartate, it will become oxaloacetate. Both are having how many carbon? 4 carbon. So this is again transamination reaction. And in this, the name of enzyme is SGOT or AST. S for serum, G for glutamate, O for oxaloacetate, T for transaminase. AST, AS for aspartate, T for transaminase. So aspartate transaminase or SGOT. You found that these amino acid means aspartate and alanine transfer its amino group to alpha ketoglutarate, and we found that 20 amino acid which take part in protein synthesis, out of 20 amino acid which take part in protein synthesis, 17 amino acid show transamination reaction, means 17 amino acid transfer their amino group to alpha ketoglutarate. So I have taken here 17 amino acid. 17 transfer their amino group to alpha ketoglutarate to form glutamate. Yes, question is asked that which amino acid don't show transamination? Mnemonic is polythene. Polythene, Po for proline, Li for lysine, thene for threonine. Yes, out of 20 amino acid, these 3 amino acid don't show transamination. We found that one amino acid transferase is delta ornithine aminotransferase. Delta ornithine aminotransferase. If this enzyme is deficient, then it lead to gyrate atrophy of retina and choroid. So you will remember this. Question has been asked, delta ornithine aminotransferase deficiency lead to gyrate atrophy of retina and choroid. Again, we come to the main topic, that is transamination reaction. Why this transamination reaction is occurring? You know, ammonia is very toxic. Ammonia is very toxic. That's why nature said that all amino acid will transfer their amino group to alpha ketoglutarate, and alpha ketoglutarate will be converted to glutamate. Now this glutamate is going, and if any other amino group is there, just like if pyrimidine ring is degraded, pyrimidine ring release ammonia, this extra ammonia will bind to glutamate and will form glutamine. Glutamine. I told you alpha ketoglutarate bind with ammonia to form glutamate. Glutamate bind with ammonia to form glutamine. Ultimately, this glutamine will travel via blood to liver. You know that ammonia is very toxic. That's why ammonia is converted to less toxic urea, and this urea synthesis or urea cycle occur in liver. So this all amino group of all amino acid are transferred to alpha ketoglutarate to form glutamate. Glutamate bind ammonia further to make glutamine. Now glutamine is a main transporter of ammonia in our body, and now this glutamine in liver will be converted back to glutamate. When glutamine will be converted to glutamate, it will release ammonia. Ammonia will go in urea cycle and will produce less toxic urea. This glutamate is further converted to alpha ketoglutarate. Again, ammonia is released and this enter urea cycle and produce less toxic urea. This last step, glutamate to alpha ketoglutarate, is catalyzed by glutamate dehydrogenase. Glutamate dehydrogenase, and you know, wherever dehydrogenase is there, hydrogen is removed, it is taken up by NAD to form NADH, or it is taken up by NADP to form NADPH. So usually dehydrogenase hydrogen acceptor is either NAD or NADP, but this is the special property of this glutamate dehydrogenase, that hydrogen acceptor may be both NAD or NADP. And this is removal of hydrogen is oxidation, removal of ammonia is deamination. This is example of oxidative deamination, and this ammonia is produced in liver and take part in urea cycle. So this is question that main transporter of ammonia in our body is glutamine. But muscle says, no, 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 I will not follow the rule. Muscle says that my main transporter would be alanine. So main transporter of ammonia in muscle is alanine. Now we come to urea cycle. I told you that ammonia is converted to less toxic form urea. Urea cycle occur in both mitochondria and cytosol. The first step occur in mitochondria. CO2 bind with ammonia to form carbamoyl phosphate by enzyme carbamoyl phosphate synthetase 1, or you can write CPS1. And in this, synthetase is there, means ATP is used. In this step, 2 ATPs are used. And we found that N-acetyl glutamate, NAG, yes, NAG is allosteric activator or obligate allosteric activator of CPS1. Frequently asked question. Obligate means it is required for the activation of CPS1. So first step in urea cycle occur in mitochondria, and it requires 2 ATP and carbamoyl phosphate is produced. Now this carbamoyl phosphate bind with ornithine. It bind with ornithine to form citrulline by enzyme ornithine transcarbamylase. Short form is OTC, ornithine transcarbamylase. This also occur in mitochondria. Now how you will remember? C, C, C. So CO2 bind with ammonia, carbamoyl phosphate bind with ornithine to form citrulline. Now citrulline come out of the mitochondria, bind with aspartate. When it bind with aspartate, it produce A4-arginino S4-succinate. So citrulline bind with aspartate to form arginosuccinate, and in this step 1 ATP is converted to 1 AMP, means ATP is converted to ADP, 1 high energy phosphate used. ATP is converted to AMP, 2 high energy phosphates are used. So I can say in this step 2 ATP used. So 2 ATP used in this step and 2 ATP used at this step. So net utilization of ATP, yes, 4 ATP used. Simple mnemonic that U looks like 4, or urea, how many letters are there? 4 letters are there. So 4 ATP used. Question is asked, sometimes they try to confuse you. 3 ATPs are involved because 2 ATP involved here and 1 ATP involved here, but how many high energy phosphates are used? 4 high energy phosphates are used. If question is asked urea cycle, how many ATP used? Best answer would be 4 ATP. And in this step where citrulline is converted to arginosuccinate, the enzyme is arginosuccinate synthetase. Synthetase ATP is used. Now this argininosuccinate is converted to arginine by argininosuccinate lysis occur. When lysis occur, this arginino component is converted to arginine, and this succinate component is converted to fumarate. Yes, we have read succinate is having 4 carbon and fumarate is having 4 carbon, and in Krebs cycle, TCA cycle, we have converted succinate to fumarate many times. So succinate and fumarate can be converted to each other. So in urea cycle, this arginino is converted to arginine and succinate is converted to fumarate. Point it out. This fumarate will enter Krebs cycle. Yes, so question is asked that in urea cycle, this fumarate is a connecting link between urea cycle and Krebs cycle. So question, connecting link between urea cycle and Krebs cycle is fumarate. Fumarate. We found that this arginine is converted to urea by enzyme arginase and produce ornithine. This ornithine enter the mitochondria. So this is urea cycle. You can easily remember 2 steps occurring in mitochondria, C, C, C. Then citrulline bind with aspartate, AS. As it will bind with AS, it will produce AS, arginosuccinate. Succinate release as fumarate, which connect to Krebs cycle, and arginine is further converted to urea and ornithine. This is urea cycle, occur both in mitochondria and cytosol. We found that pyrimidine synthesis occur in cytosol and mitochondria also, but initial steps occurring in cytosol. So this pyrimidine synthesis, glutamine bind with CO2 to form carbamoyl phosphate by CPS2. So question is asked that CPS1 is used in urea cycle. CPS2 is used in pyrimidine synthesis. CPS1 is present in mitochondria. CPS2 is present in cytosol. So this step of pyrimidine synthesis occurring in cytosol. So this carbamoyl phosphate is further converted to orotic acid. This is pyrimidine synthesis. So we know that carbamoyl phosphate can be converted to orotic acid. Now question is asked that if CPS1 is deficient, carbamoyl phosphate synthetase 1 is deficient, then it is called hyperammonemia type 1. Means CPS1 is not there, then ammonia will not be detoxified, ammonia will increase, hyperammonemia type 1. If OTC is deficient, then this carbamoyl phosphate will not be metabolized. So this carbamoyl phosphate will increase. Carbamoyl phosphate will increase and ammonia will increase. When ammonia will increase, that is called hyperammonemia type 2. So hyperammonemia type 2 OTC is deficient. And we found that in OTC deficiency, this carbamoyl phosphate will accumulate. This will go to cytosol and will enter the pyrimidine synthesis, and this carbamoyl phosphate will produce orotic acid, more and more orotic acid. So in OTC deficiency, orotic acid urea. So you will find catching point in urea cycle defect. If orotic acid urea is there, this is hyperammonemia type 2 clinical scenario questions are asked. And this hyperammonemia type 2 is most common urea cycle defect. It is X-linked recessive and X-linked partial dominant. At few places it is written X-linked partial dominant. So it is X-linked recessive, so more common in male than female. So these two urea cycle defects are important, hyperammonemia type 1 and hyperammonemia type 2. Type 2 is more important. I told you that in urea cycle defect, ammonia will increase and ammonia is very toxic. Why it is toxic? We found that ammonia bind with alpha ketoglutarate. I told you alpha ketoglutarate will be converted to glutamate. And this glutamate bind with further ammonia to form glutamine. So if ammonia is very high, then this alpha ketoglutarate will be converted to this glutamine, and this will be decreased. And when this will decrease, Krebs cycle intermediate will be depleted. If this Krebs cycle intermediate is depleted, Krebs cycle will not occur. Question is asked that in urea cycle defect, which intermediate of Krebs cycle is depleted? So in urea cycle defect, alpha ketoglutarate is depleted. Question. And when this is depleted, Krebs cycle will not occur. And brain is very dependent on Krebs cycle. So brain will not be able to do function properly. And this will lead to hepatic encephalopathy, means liver will not be able to detoxify ammonia. Ammonia will interrupt this Krebs cycle and will lead to encephalopathy because brain is dependent on this Krebs cycle, hepatic encephalopathy. We found that aspartate and oxaloacetate, the only difference is of ammonia. So if I join amino group 2 oxaloacetate, it will be converted to, point it out, it will be converted to aspartate. Yes, oxaloacetate and aspartate, the only differences of ammonia. If I join ammonia to oxaloacetate, it will convert to aspartate. So question was asked, connecting amino acid, point it out. If they ask connecting link, then answer is fumarate. But they ask connecting amino acid between urea cycle and Krebs cycle, that is aspartate. And we know urea is NH2CO NH2. NH2CO NH2, And question is asked that this nitrogen comes from, or ammonia contribute this nitrogen, this CO2 contribute this CO, and the second nitrogen is contributed by aspartate. So in NH2CO, NH2, nitrogen comes from ammonia, CO comes from CO2, second nitrogen comes from aspartate, mnemonic aspartate participate. And what is the treatment of urea cycle defect? If hyperammonemia is there, we have, yes, scavenger of nitrogen, that is phenyl butyrate, which is prodrug, which is converted to phenyl acetate. Yes, phenyl acetate bind with glutamine to form phenyl acetyl glutamine, which is excreted in urine, water soluble. So in this way, yes, this phenyl butyrate is used in urea cycle defect. Now we come to structure of protein. Suppose I give you a protein having 4 amino acid and I ask you what is the primary structure of protein. I gave you this protein having 4 amino acid and I ask you what is the primary structure of this protein. You will tell, sir, this protein is having 4 amino acid. First is alanine, second is leucine, third is valine, fourth is lysine. You told me sequence of amino acid means primary structure of protein means sequence of amino acid in a protein. And we found that in a protein, these amino acids are joined by amide bond, peptide bond, and these amide or peptide bonds are covalent bond. So question is asked that primary structure means sequence of amino acid, main bond is covalent bond. It is stabilized by covalent bond. Now, suppose a protein is having 1000 amino acid. These 1000 amino acid will not remain in a linear chain. They will fold themselves. Yes, they may fold themselves in a helical structure. If it is helical, then it is called alpha helix. Sometimes they will fold themselves or sequence of amino acid will fold on this sheet-like structure that is called beta sheet. So we found that this sequence of amino acid is folding. If folding of the sequence, then it is called secondary structure. If this folding is just like a sheet, then it is beta sheet. Means this sequence is making a sheet-like structure, beta sheet. So secondary structure, we found that 2 examples of secondary structure which are commonly asked. Secondary structure may be alpha helix or beta sheet. If sequence of amino acid fold in a helical form, that is alpha helix. Okay, and if it fold in a sheet-like structure, this is beta sheet. And we found that this secondary structure is stabilized by hydrogen bond. Stabilized by which bond? Hydrogen bond. So question is asked, secondary structure, main bond is hydrogen bond. We found that secondary structure may be alpha helix or beta sheet. And we found that proline is never found in alpha helix. This is question. Except position 1. Means at position 1, proline may be there in alpha helix, but it is not found other than first position. Proline is never found. Question is asked. Glycine is usually not found in alpha helix. Question, how questions are asked, they will tell you that there is a protein in which alpha helix is disturbed. Which amino acid will disturb this alpha helix? Answer would be proline. Best answer. If proline is not given, answer would be glycine. So we have discussed primary structure and secondary structure. We found that secondary structure, suppose this is beta plate sheet and it can't do function. Means suppose this protein is having this beta sheet secondary structure and want to carry this cap. Suppose the function of this protein is to carry this cap. It can't do function. But if you fold this secondary structure further, now it is tertiary structure. Tertiary structure means folding of secondary structure, that is tertiary structure. And if you put this cap here, then now it can carry. Means primary structure, sequence of amino acid. This sequence of amino acid fold, they form secondary structure. This folded beta sheath further fold and now protein can do function, and this is called tertiary structure. So tertiary structure, protein can do function. This question is asked. So now, in primary and secondary structure, functional activity is absent. But tertiary structure, Functional activity is present. Functional activity is present. Main bond of tertiary structure is disulfide bond. This question is asked. So primary structure covalent bond, secondary hydrogen bond, and tertiary structure main bond is disulfide bond. Now, more than one functional polypeptide chain is called quaternary structure. More than one functional polypeptide chain, just like hemoglobin. You know, hemoglobin is having 4 globin chain, and each globin chain can bind to 1 molecule of oxygen. Means each globin chain can do function. Or you can understand like this. This is tertiary structure which can do function, can carry cap. I took one more beta plated sheath and again fold. And now this is again tertiary structure, and now this can do function. If I take these two tertiary structure and join together, means this is quaternary structure. Quaternary structure means more than one functional polypeptide chain, just like hemoglobin. 4 functional polypeptide chains are there. So hemoglobin is having quaternary structure also. So quaternary structure main bond is hydrophobic bond. This is question. And hemoglobin is having quaternary structure. But we found myoglobin is having only one functional polypeptide chain. So it can bind to only 1 molecule of oxygen. So it is having tertiary structure, but no quaternary structure. Quaternary structure means more than one functional polypeptide chain. But myoglobin is having only one polypeptide chain. So no quaternary structure in myoglobin. We found that functional activity is present in tertiary and quaternary structure, and if I heat the protein, protein is denatured. If you heat a protein, protein is denatured, and when you heat a protein, this secondary, tertiary, quaternary structure is lost. But primary structure, that covalent bond, peptide bond, is not broken down by heat. So this is retained. Question is asked that on denaturation of protein, protein can't do function. In denaturation, only primary structure remain intact. These, yes, secondary, tertiary, quaternary structure are lost. These are lost. Question is asked on detection of primary structure can be done by mass spectrometry and advanced technique. Best is mass spectrometry. This is best for primary structure. One question on mass spectrometry, that mass spectrometry can also determine molecular weight of protein. So it can determine primary structure, means sequence of amino acid. It can also determine molecular weight of protein. Secondary, tertiary, quaternary structure are detected by X-ray crystallography, NMR spectrometry. Best is X-ray crystallography. But now cryo-electron microscopy is becoming popular. I will tell you line from Harper and you will decide according to that in questions. Cryo-electron microscopy is rapidly superseding X-ray crystallography as method of choice for determining 3-dimensional structure of protein and other biological macromolecule. I have given you these lines. When question would be asked, they will definitely give some hint and modified language. You will be able to recognize this. Cryo-electron microscopy. A question is asked, most accepted method for A1C estimation. 1. Yes. I. Looks like I. Ion exchange chromatography. Most accepted method of HbA1c is ion exchange chromatography. Now we come to 21st amino acid, that is seleноcysteine. Seleноcysteine is 21st amino acid and it is produced by co-translational modification. This is question. You know that there are 20 amino acids which take part in protein synthesis, but we found that there is 21st amino acid, that is seleноcysteine, which also take part in protein synthesis. And how it is produced? From which amino acid it is produced? We found that precursor for seleноcysteine is a serine. How you will remember? Se for serine, Se for seleноcysteine. So we found that serine is converted to seleноcysteine and this is produced by co-translational modification. And during protein synthesis, we know there are 3-stop codon, UAA. 3-stop codon are UAA, UAG, UGA. You are away, you are gone, you go away. These are 3-stop codon. Whenever these stop codon comes, protein synthesis stops. But we found that this seleноcysteine, yes, recognize stop codon UGA. And where UGA is there, this selenocysteine will be attached. So this is recoded. So question is asked that which stop codon code for selenocysteine, that is UGA, mnemonic is yoga. Selena Jetli or Selena Gomez is doing yoga. And question is asked that which enzymes are having selenocysteine. So when she is doing yoga, you are observing gluteal region, glutathione peroxidase, thigh region, thyrodoxine reductase, G for glutathione, G for glycine reductase. Yes, thyrodoxine reductase means these three you can easily remember by gluta word. Yes, deiodinase and selenoprotein P. These are the enzyme having selenocysteine. Now, protein folding chaperone. Suppose this is a protein having tertiary structure. You heat this protein. This protein is denatured. Denatured. Now this can't do function. When this can't do function, now protein will be tried to be repaired. Means chaperone will come, chappy will come and will try to refold it normally. So chaperone do folding of protein. So I told you if heat comes, protein is denatured and this chaperone will try to correct it. That's why chaperone is also called heat shock protein. And in this folding, yes, when it will try to fold it, yes, it will use, if you take iron rod and try to fold it, you will require energy. In the same way, chaperone will try to fold protein again and will require energy. So one question, chaperone use ATP. Use ATP. So chaperone folded it correctly. Suppose this is denatured protein, chaperone came or heat shock protein came and it corrected it. Now it will do the function. So corrected, so it will do function. But if it is not corrected, then chaperone will attach it with ubiquitin. Yes, now this misfolded protein attached with ubiquitin, this unit will go to proteasome. And proteasome will degrade this protein. Why? Because chaperone has tried to correct it, it is not corrected, to better to degrade. How this proteasome will know that this protein should be degraded or not? If it is attached with ubiquitin, yes, ubiquitin, quit karo abhi. Abhi quit karo means degrade it. So proteasome will degrade only those misfolded protein which is attached with ubiquitin. Ubiquitin is the trademark. Yes, I have tried, but it could not be repaired. Better to degrade. So proteasome degrade. Yes, ubiquitin attached misfolded protein and amino acids are released. Now few tests. Hartnup disease, we do Obermeyer test. Mnemonic is obey your heart. Dil ki mano, Obermeyer, obey heart, Hartnup disease. Homocystinuria, for homocystinuria, we do cyanide nitroprusside test. How you will remember cyanide, CY for cyanide, CY cyanide, NI if you read backward, CY for cyanide, NI for nitroprusside. So homocystinuria, cyanide nitroprusside test. For pheochromocytoma, we do VMA spot test. Mnemonic is WIMBAR. Yes, if you are using WIMBAR, there would be pheo means feca, chromo means color. Color would be feca. So feca color, that is pheochromocytoma by WIMBAR VMA spot test. Recently asked question on fish odor disease, in which there is deficiency of trimethylamine oxidase. Trimethylamine oxidase deficiency lead to trimethylaminuria. This is also called fish odor disease. And in this, symptoms will be aggravated or increased by taking choline. So if you take choline, symptoms will increase in fish odor disease. Recently asked question. Now we are going to discuss amino acid as deuter ion. Deuter ion have both positive and negative charge in equal amount. What does it mean? Whenever you see amino acid, it is having amino group, it is having acidic group. And you know, wherever amino group is there, it will be ionized to acquire positive charge. Ionized means one ionizable group is amino group. Carboxylic group, every time it is ionized to acquire negative charge. So This second ionizable group is carboxylic group. So I can say that amino acid is having two ionizable group, that is amino and carboxylic group. So ionizable groups are having ionization constant. So ionization constant pK is ionization constant. I can say that amino acid is having two ionizable group. That's why amino acid is having two ionization constant pK1 and pK2. We found that amino acid has net charge 0 on a particular pH. Means at a particular pH, net charge on amino acid or Jupiter ion or protein is 0. That is called isoelectric point. So isoelectric point is the pH at which net charge on protein is 0. When net charge is 0 means it would be least soluble. When charge would not be there, it would be least soluble and maximum precipitation would occur. So if you don't remember this detail, it's okay. But remember that amino acid is having two ionizable group and there is a concept of isoelectric point that is called Pi. If you want to calculate isoelectric point of a particular Jupiter ion or an amino acid, what you can do? You will do average of pK1 and pK2. You will find the Pi. Simple numerical question is asked that pK1 means ionization constant of one group is 2. Ionization constant of another group is 10. What is the isoelectric point? So you will take the average of 2 plus 10 divided by 2, 6. So if they have given two ionization constant and they ask isoelectric point, you will take the average of these two. That would be the Pi, that is isoelectric point. Is the pH at which net charge on protein is 0. Now, buffer. What is buffer? Buffer resists change in pH on adding acid or alkali. Suppose this is a solution. In this solution, there is CH3COOH. This is weak acid. Means carboxylic group is there. If carboxylic group is this is ionizable group. Ionizable group. Yes, you know, wherever carboxylic group is there, it ionizes to acquire negative charge. We found that this carboxylic group, ionizable group, is having a particular pK, ionization constant. If you start adding alkaline in it, alkaline, as you know, alkaline, you will add, pH will increase, but this carboxylic group will resist this change. That is called buffering capacity. Yes, buffer resists change in pH on adding acid or alkaline. And we found that buffer is most effective at pH equal to pK, ionization constant. Means now titration curve. What is titration curve? Before going to definition, look at this. I took this OH, means addition of alkali at x-axis, and I took change in pH on y-axis. Now, in this solution, I am adding alkali. As I will add alkali, this pH will increase. And we found that after adding alkali at a particular point, if you add further alkali, pH is not changing. pH is not changing. So if pH is not changing, means this is the buffer capacity of this carboxylic group. Yes, this acetic acid is having a buffering capacity, means it resists the change in pH. So we found that at this point, there is no change in pH. We can say at this point, there is a maximum buffering capacity and buffer is most effective at pH equal to pK. So I can say this B point, at B point, there is no change in pH on adding alkali. Yes. So maximum buffering capacity is at B point. And you know, pK is the pH at which buffer is most effective. So you can say B point is pK. So for ionization constant, pK value, yes, you can find this titration curve in which B point is pK. ionization constant. Now, what is titration curve? No need to remember the definition, but just have an idea. Graphic plot of pH of buffer against amount of alkali added. So graphic plot between OH and pH. And at this point, no change in pH. So this is ionization constant. So first of all, sole question. If question is asked, they have given this question previously also, they will give you this graph and they will ask you, yes, how many ionizable groups are there? Only one ionizable group is there. If they give this graph, you will see two ionizable groups are there. Yes, if they give this graph, you will see three ionizable groups are there. And ionizable group have ionization constant. So this is having pK1, pK2, pK3. This is having pK1, pK2. So now I explain. Titration curve of weak acid having one ionizable group, example carboxylic acid, and in this we found only one pK. Yes, because only one ionizable group, and at this pK there would be maximum buffering capacity. Okay, this is titration curve of amino acid having two ionizable group. Yes, 2 pK value. And if you want to determine that isoelectric point, what you will do? You will take the average of pK1 and pK2. If this is the average, this is the C point. C point is isoelectric point. And this is the titration curve of amino acid having three ionizable group. So I conclude this is complicated topic. Sometimes questions are asked in which they will give you graph and they will ask how many ionizable groups are there. Yes, you will see that this one ionizable group, this two ionizable group, this three ionizable group. So if one ionizable group, 1 pK, means at this point maximum buffering capacity. If two ionizable group, 2 pK, on these 2 points, maximum buffering capacity. If 3 ionizable point, 3 pK, at these 3 points, maximum buffering capacity. If they ask what is isoelectric point, you will take the average of pK1 and pK2 and you will tell them this is the isoelectric point. And you can tell that how many ionizable groups are there in this titration curve. Now we start molecular biology or genetics. We know that DNA, RNA is made up of purine and pyrimidine base. Purine base mnemonic is pure as gold. A for adenine, G for guanine. So if you find a structure having 2 rings, you will say this is purine ring. And if purine ring is given, how you will identify that this is adenine or guanine? You will search for amine group. If only amine, then it is adenine. If structure is given, you find 2 rings are there and you find that amine is there and keto group, both are there, then it is guanine. So if 2 rings are there, this is purine, pure as gold, adenine or guanine. If only amine group is there, that is adenine. If amine and keto group both are there, that is guanine. These are purine bases. Pyrimidine bases are having only single ring, mnemonic is cut. C for cytosine, U for uracil, T for thymine. So pyrimidine is having only 1 ring. If you find 1 ring, you will search again for amine group. If amine group is there, this is cytosine. And we have read, whenever you remove amino group, there is joining of keto group. So if I remove amino group by enzyme cytosine deaminase, this cytosine would be converted to uracil, means this amine group will be converted to keto group. So only difference between cytosine and uracil is amine group and keto group. And we found that in our body, cytosine can be converted to uracil by cytosine deaminase. If I see the structure of this thymine is exactly uracil with additional methyl group. Name itself tells you methyl. If methyl group is there, that is thymine. So you can compare the structure. This is same. This keto group is same. Only methyl group is extra. So if you want to convert uracil to thymine, you must add methyl group. And methyl group donor is folic acid tetrahydrofolate. So question was asked that deamination followed by methylation of cytosine will produce. So deamination followed by methylation of cytosine will produce, yes, thymine. If folic acid is not there, then uracil will not be converted to thymine. We know that DNA is having double stranded structure, A, T, 2 hydrogen bond, G, C, 3 hydrogen bonds are there, and in RNA there is A, U, G, C. So if folic acid is not there, then uracil will not be converted to thymine. So misincorporation of uracil into DNA is seen in deficiency of folic acid. If folic acid is deficient, uracil will not be converted to thymine, and in place of thymine, DNA may have uracil. So these are base, purine base and pyrimidine base. We found that base plus sugar nucleoside, base plus sugar plus phosphate nucleotide. 3 component, base, sugar, phosphate, nucleotide. 2 component, S4 sugar, S4 side. Synthesis of purine and pyrimidine require. We found that purine synthesis require aspartate, glutamine, CO2, glycine, and folic acid. In pyrimidine, we require aspartate, glutamine, CO2. So I can say initial 3 are same. Yes, aspartate is common to both, glutamine is common to both, CO2 is common to both. So purine, INE, pyrimidine, INE, that is glutamine, not glutamate. Many times question has been asked that which of the following is not contributor of purine ring. So aspartate participate, glycine, yes, glycine participate, glutamine, yes, glutamine, INE, glutamine, not glutamate. Answer is D. It is not glutamate which contribute. Who contribute? Glutamine contribute, not glutamate. Now this is a strand of DNA. In this strand of DNA, this is 5' and this is 3' end. We found that DNA and RNA, the differences are DNA is usually double-stranded DNA, RNA is usually single strand. DNA A bind with T, 2 hydrogen bond, GC, 3 hydrogen bond, while RNA A, U, G, C means DNA is having T, RNA is having U. And DNA is having deoxyribose sugar while RNA is having ribose sugar. So what is the difference between ribose and deoxyribose sugar? If you see the structure of this ribose sugar, there is OH at C3 and OH at C2. In deoxyribose, OH is present at C3 but no oxygen at C2. Point it out. Deoxy, removal of oxygen at C2, deoxyribose. If no oxygen at C2 and C3, dideoxyribose. So dideoxyribose, deoxyribose, ribose is present in RNA, deoxyribose is present in DNA, dideoxyribose don't take part in DNA synthesis. Dideoxyribose inhibit DNA synthesis. Before that, I will discuss that this is a strand of DNA. Suppose this is a strand of DNA. We found that in this OH group of C3 bind with OH group of C5 and this is phosphodiester bond. Phosphate bind with 2 OH to make phosphodiester bond, and question is asked that backbone of DNA or backbone of RNA is stabilized by phosphodiester bond. So question, backbone of DNA is stabilized or RNA is stabilized by 3,5 phosphodiester bond. Phosphodiester bond. DNA synthesis always occur in 5' to 3' direction. Question. We always read in 5 to 3 direction. Synthesis always occur in 5 to 3 direction. We always write in 5 to 3 direction. And there should be OH group at C3 of sugar. Yes, at C3 of sugar, there should be OH to make phosphodiester bond. As you know, phosphodiester bond is formed between OH of C3 and OH of C5. So because new nucleotide join by 3,5 phosphodiester bond. Okay. In dideoxynucleotide, there is no OH at C3. Yes, I told you dideoxynucleotide, no OH at C3. So if dideoxynucleotide added in PCR, DNA synthesis, this PCR will stop. PCR will stop. So dideoxynucleotide or dideoxyribose is not having OH at C3, and when OH is not present at C3, you know that phosphodiester bond is formed between OH group of C3 and OH group of C5. So dideoxyribose don't take part in DNA synthesis. So why we are using it? Because there is a technique that is called chain termination method or Sanger sequencing, which is used in determining the DNA sequence. If I give you a DNA sequence and I ask you to determine what is the nucleotide sequence, you can determine by a technique that is called Sanger sequencing or chain termination method. In this technique, we use dideoxynucleotide. So in Sanger's technique, dideoxynucleotide is used because it does not allow further elongation of DNA synthesis. Means it inhibits DNA synthesis. Because DNA synthesis, there should be OH at C3. I just told you Watson and Crick base pairing rule. A bind with T, 2 hydrogen bond. G bind with C, 3 hydrogen bond. So they gave this structure, complicated structure, and they ask what is the marked bond. This is hydrogen bond. Between AT, 2 hydrogen bond. Between GC, 3 hydrogen bonds are there. So answer is hydrogen bond. If they ask what is this bond, phosphate make phosphodiester bond. So this is phosphodiester bond. If they ask sugar or base bind with, so here it's not so clear, but sugar and base, this is sugar and base bind with N-glycosidic linkage, N-glycosidic linkage or beta N-glycosidic linkage. Next time they can ask question in this diagram itself, and you will identify very easily that this is nitrogen and this is pentose sugar. So this is N-glycosidic linkage. Now question is asked on Chargaff rule. Chargaff rule is A plus G is equal to T plus C. Conditions should be that this rule is applied in double-stranded DNA. You know A bind with T, G bind with C. So A and G jointly would be equal to T and C, T plus C. And concentration of A would be equal to concentration of T. Concentration of G would be equal to concentration of C. So if they give concentration of any one nucleotide, you can calculate the another. Just like they ask question that thymine residue in DNA is 20%, 28% T. So A would be 28%. So A plus T is equal to, yes, 56%. Total 100% minus 56%, there would be 44% would be G plus C. And you know the concentration of G is equal to concentration of C. So you can say 2C is equal to 44. So C is equal to 44 divided by 2, that is 22%. Simply you can calculate if A concentration is given, that would be equal to T, 28%. G would be equal to C. Total percentage would be 100%. So 22%. This question has been asked about Chargaff rule applied on double-stranded DNA. Now, purine catabolism and pyrimidine synthesis. Purine, that is AMP, GMP. Yes, adenosine monophosphate, guanosine monophosphate. AMP is converted to adenosine. Adenosine is converted to inosine. This adenosine is converted to inosine by adenosine deaminase, ADA, adenosine deaminase. If adenosine deaminase is deficient, this enzyme is deficient, this lead to severe combined immunodeficiency. How you remember mnemonic CID? CID ne chapa mara on ADA of, yes, DACUs. So ADA, ADA, SCID. We found that inosine is converted to hypoxanthine, guanosine is converted to guanine. This hypoxanthine and guanine are converted to xanthine by xanthine oxidase. This xanthine is converted to uric acid by the same enzyme xanthine oxidase. So question is asked, end product of purine catabolism is uric acid. The uric acid is further converted to allantoin by enzyme uricase. This is in non-primate mammals. We developed RASP uricase, that is recombinant form of uricase, which is used in tumor lysis syndrome. In tumor lysis syndrome, tumor cell is degraded and more purine are catabolized, more and more uric acid is produced. We convert this extra uric acid to allantoin, which is more water soluble and excreted easily. So recombinant form of uricase is RASP uricase, which is used in tumor lysis syndrome. Allopurinol is a drug which inhibits xanthine oxidase. Clear? So this is about purine catabolism. Purine synthesis may be de novo synthesis or synthesis by salvage pathway. Means when these purines are being degraded, at that time these purine can be saved from degradation with the help of enzyme HGPRT. So we found that in our body, salvage pathway is present in brain, RBC and WBC, in which purines are prevented from degradation with the help of enzyme HGPRT. HGPRT convert guanine to GMP, hypoxanthine to IMP. If this HGPRT is completely deficient, it lead to Lesch-Nyhan syndrome. In Lesch-Nyhan syndrome, there is self-mutilation, yes, self-injurious behavior, neuropsychiatric symptom, and gouty symptoms would be there. Means if this step is not occurring, means if this is not converted back, then guanine and hypoxanthine, more and more guanine and hypoxanthine will be converted to uric acid, hyperuricemia, gouty symptoms would be there. Lesch-Nyhan syndrome is X-linked recessive disorder. Question. So more common in males. So Lesch-Nyhan syndrome, frequently asked question, they will give you this image and they will say that there is hyperuricemia, and the treatment part we give allopurinol because allopurinol inhibit xanthine oxidase, decrease uric acid synthesis, gouty symptoms are improved. A question is asked, salvage pathway is utilized in hybridoma technology. This is recently asked question. Now pyrimidine synthesis. In pyrimidine synthesis, glutamine bind with ATP plus CO2 to form carbamoyl phosphate by CPS2, carbamoyl phosphate synthetase 2. This carbamoyl phosphate is further converted to orotic acid by multiple steps. This orotic acid is converted to OMP by orotate phosphoribosyl transferase. OMP is converted to UMP by decarboxylase. Question is asked, first pyrimidine nucleotide to be synthesized is U, and this U is further converted to C and T, and you know that T is present in DNA, so it should be deoxy. So DUMP is converted to DTMP by thymidylate synthase. This thymidylate synthase is inhibited by 5-fluorouracil. Question. So 5-fluorouracil inhibit thymidylate synthase. And we found that when this DUMP is converted to DTMP, you know what is the difference between U and T? I have discussed that U and T, the only difference is methyl group, and methyl group donor is folic acid. So we found that N5 and 10 methylene tetrahydrofolate is converted to dihydrofolate in this reaction. Means methyl group donor is N5 and 10 methylene tetrahydrofolate. This dihydrofolate is converted back to tetrahydrofolate by dihydrofolate reductase. And this dihydrofolate reductase is inhibited by methotrexate. This recently asked question, and in NEET PG24 they asked question that methotrexate reduce synthesis of DTMP. So if methotrexate is there, this step would not occur, this step would not occur, so DTMP will not be synthesized. We found that if this orotate phosphoribosyl transferase or decarboxylase is deficient, this lead to hereditary orotic aciduria. Why? If this step is not occurring, this orotic acid will not be metabolized. When orotic acid not be metabolized, orotic aciduria would be there. And if orotic aciduria is there, this UMP would not be synthesized, TMP would not be synthesized, CTP would not be synthesized. So means nucleic acid will not be synthesized, means cell will not be able to divide, but cytoplasm will grow. So macrocytic anemia. In macrocytic anemia, most of time you will find that they respond to B9 folic acid and B12, but in hereditary orotic aciduria will not respond to B9 or B12. In hereditary orotic aciduria, if I want to treat the patient and you are asked that give either U, C or T, which you will prefer? You will say, sir, I will give U because we know that the first which is synthesized, that is U. We have defect at this level. If I bypass this and I give U, U will be further converted to C and T very easily. So in hereditary orotic aciduria, I will give U and C and T will be synthesized easily. Product of catabolism of nitrogenous base, purine, I told you, and product is uric acid. Pyrimidine, C and U, they are degraded to produce CO2, ammonia, and beta alanine. While thiamine, yes, T, is degraded to CO2, ammonia. These two are same, CO2 and ammonia are same, and beta amino isobutyrate. Now we come to replication of DNA, double-stranded DNA. You know, two strands are anti-parallel, 5 end is against 3 end, 3 against 5 end. In double-stranded DNA, two strands are separated and each strand acts as template and new strand is synthesized. So in DNA synthesis or DNA replication, first of all, these two strands should be separated, and that is done by helicase. Helicase separate double-stranded DNA, and in this it use ATP, so ATP used. We found that when double-stranded DNA is separated, there is production of supercoiling, torsional tension. That torsional tension is removed by next enzyme topoisomerase, remove supercoiling. Means two strands are separated. Now each strand will act as template, but we know in DNA synthesis, before DNA synthesis, there must be a primer. Means DNA synthesis occur by attaching new nucleotide to primer. So before DNA synthesis, we must synthesize primer. We found that when two strands are separated, one strand act as leading strand, another strand act as lagging strand. Why leading? On one strand, synthesis occur continuously. If synthesis occur continuously, will be completed early, that strand will lead, that's why leading strand. On another strand, synthesis occur in small fragments. When synthesis occur in small fragments, it will take more time and it will lag. That's why it is called lagging strand. And I just told you that every time before DNA synthesis, we require primer. So in leading strand, you require only one primer and then continuous synthesis will occur. But on lagging strand, before each fragment synthesis, there would be primer. So on lagging strand, multiple primer are there and synthesis occur in small fragments, and these small fragments are called Okazaki fragments. So now I say that next enzyme is primase. Primase synthesize primer, but primer is not DNA, primer is RNA. Point it out. Now, when primer is synthesized, now I can do DNA synthesis. Next enzyme is DNA polymerase 3. DNA polymerase 3 will come and will synthesize complete leading strand. Means on leading strand, we attach only one primer, and after attaching this primer, this DNA polymerase 3 will synthesize complete leading strand continuously. Leading. That's why it is leading. It is doing the work continuously. The same DNA polymerase 3 synthesize small fragments on a lagging strand. These small fragments are called Okazaki fragments. So question is asked that Okazaki fragments are synthesized by DNA polymerase 3 and they are synthesized on a lagging strand. So I can say that DNA polymerase 3 synthesize complete leading strand continuously and small fragments known as Okazaki fragment on lagging strand. Now, this is not complete because I told you these primer are RNA. These primers are RNA and we are doing DNA synthesis. So these primers should be removed. So next function is primer removal that is done by DNA polymerase 1. Remove primer and fill the gap. Means DNA polymerase will remove primer by 5 to 3 exonuclease activity. 5 to 3 exonuclease activity. What does it mean? We found that suppose this is DNA strand. This is 5 end, this is 3 end. If I cut from the end, yes, if I start cutting from this end and move in this direction, I will say this is 5 to 3 exonuclease cutting from the end. If suppose this is DNA strand and I cut from the middle, that is endonuclease. Endonuclease cut from the middle. Okay, and here and exonuclease cut from the end. So 3 to 5 exonuclease will cut from 3 end and will move towards 5 end. 5 to 3 exonuclease will move from 5 to 3 direction. This question is asked. So primer removal, yes, is 5 to 3 exonuclease activity. Means DNA polymerase 1 will come and will remove primer in this direction, 5 to 3 exonuclease activity. So on the lagging strand again, primer will be removed by DNA polymerase 1. DNA polymerase 1 will remove primer from 5 to 3 end. And this gap will be produced. This gap is filled by DNA polymerase 1 itself. Now, on the lagging strand, you made Okazaki fragments, new fragment, Okazaki fragment. These new fragment and Okazaki fragments are joined by DNA ligase, ligate. And it act only on lagging strand. Ligase, mnemonic ligase act only on lagging strand. Now, question is asked that ligase require ATP. First question, it make phosphodiester bond. So question has been asked on the sequence, first helicase, then topoisomerase, then primase, DNA polymerase 3, DNA polymerase 1 and DNA ligase, sequence of enzyme in DNA replication. Now, in prokaryotic DNA polymerase, I told you DNA polymerase 1, 2, 3 and I told you 3. It synthesized complete leading strand and small fragments called Okazaki fragments on lagging strand. DNA polymerase 2 do DNA repair. DNA polymerase 1, I told it remove primer and fill the gap. And DNA polymerase 1 has also role in DNA repair. So this is about prokaryotes, named as 1, 2, 3. But in eukaryotes, polymerase are named as alpha polymerase, beta, gamma, delta, epsilon. So how you will remember? Yes, which polymerase do which function? First of all, out of these gamma, Ma means mitochondria. So gamma polymerase do mitochondrial DNA synthesis. In our cells, mitochondria is having double-stranded DNA, double-stranded circular DNA. No introns in mitochondrial DNA. So introns are not there, circular double-stranded DNA is there, and due to absence of intron, mutation rate is high in mitochondria than nuclear DNA. Point it out. Now, alpha polymerase synthesize primer. So primase activity, alpha polymerase has primase activity. Beta polymerase do DNA repair. Gamma mitochondrial DNA synthesis. We found that in eukaryotes, leading strand is synthesized by epsilon, L-E-A-D-I-N-G. Leading strand is synthesized by epsilon. Lagging strand, if you revert, yes, delta, yes, it looks like G. So lagging strand is synthesized by delta polymerase. We found that all these polymerase I have discussed, all these polymerase have proofreading, except alpha and beta polymerase. Alpha and beta polymerase, no proofreading. What is proofreading? Proofreading means 3 to 5 exonuclease activity. Yes, remember, primer removal means 5 to 3 exonuclease activity. Yes, proofreading 3 to 5 exonuclease activity. So how you will remember? Primer person, prime person, higher level to lower level. So 5 to 3 exonuclease make any mnemonic, and proofreading 3 to 5 exonuclease activity. All polymerase have proofreading 1, 2, 3, gamma, delta, epsilon. Out of these 8, 6 are having proofreading except alpha and beta polymerase. Question is asked. Now, DNA polymerase 1, 2, 3 all can synthesize DNA, and DNA synthesis always occur in 5 to 3 direction. So all these 3 have 5 to 3 polymerase activity, definitely. We found that all polymerase, I told you 1, 2, 3, all these 3 have proofreading. So all these 3 have 3 to 5 exonuclease activity. I told you proofreading is 3 to 5 exonuclease activity. Yes. We found that DNA polymerase 1 has extra function, that is 5 to 3 exonuclease activity, primer removal. So this enzymatic activity is 5 to 3 exonuclease activity, primer removal, which is specific to DNA polymerase 1. Question is asked. A question is asked on cleano fragment. Cleano fragment is DNA polymerase 1 from which this part is deleted. Means DNA polymerase 1 in which primal removal activity is lost, or you can say DNA polymerase 1 minus 5 to 3 exonuclease activity. So this image will help you to solve many questions recently asked. Now, telomere. We found that in our body, in chromosome, at linear end, we found there is a sequence that is TTAGGG. TTAGGG. First question, telomere, what is the sequence? Sequence is TTAGGG. Tail, GGG. Yes, naughty? Yes. Telomere sequence, these are present at linear chromosome end. So present at end of linear chromosome. Some part lost after each replication. We found that after each replication, small portion of DNA is lost, and nature is very wise. This telomere is not going to code for any protein. That's why nature kept telomere in the end of chromosome. So whenever DNA will divide, the lost portion would be telomere, and telomere is not going to affect any protein. So after each replication, a small portion of DNA is lost, that is telomere. So in somatic cell can divide up to a limit. When all telomere will be lost, then somatic cell will not be able to divide further, and this limit is called Hayflick limit. Question is asked that due to shortening of telomere, a somatic cell can divide up to a limit that is usually 40 to 60 times, and this limit is called Hayflick limit. But in germ cell, stem cells, germ cells, stem cells are dividing rapidly, regularly, but they don't die. Why? Because germ cell and cancer cell, there is an enzyme, telomerase enzyme. Telomerase enzyme, when you see the structure of telomerase enzyme, you will find it is having one RNA template and polymerase. So I show you. This is linear end of chromosome, TTAGGG, 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 this telomere is there. And after each replication, this will be shortened. And we have enzyme telomerase. This is telomerase. Telomerase has two components. One component is RNA template. This is RNA template. And second component is this polymerase. So this RNA template goes to DNA, and with the help of polymerase, means this template go, and with the help of this polymerase, this DNA synthesis occur. So DNA synthesis occur on this RNA template. So RNA dependent DNA polymerase. So I can say telomerase synthesize DNA from RNA template. So I can say that telomerase is RNA dependent DNA polymerase. Point is important that telomere enzyme has both RNA template and protein polymerase. and this is reverse transcriptase. Reverse transcriptase. Yes, RNA dependent DNA polymerase or reverse transcriptase. We found that as you are going older and older, means aging, telomerase activity is decreased. In progeria syndrome, yes, premature aging, PA movie, Amitabh Bachchan, that telomerase activity is decreased. In cancer cell, yes, cancer cell divide regularly, don't die, because we found that in cancer cell telomerase activity is increased. Question: telomerase activity increase in cancer, decrease in aging, decrease in progeria syndrome. This is RNA dependent DNA polymerase, also called reverse transcriptase. Hayflick limit. Question: now DNA repair mechanism and disorder due to their defect. So we found that repair mechanism, double-stranded break repair mechanism include two. Double-stranded break, DNA, double-stranded DNA, double-stranded break occurred. Yes, it is repaired by non-homologous end joining or homologous recombination. If non-homologous end joining repair defect is there, it lead to severe combined immunodeficiency, SCID. Mnemonic is CID, tiger. Tiger is having no home. During that movie we saw, he was wandering. CID, row agent, same. So no home, non-homologous. And homologous means home. Anything related to home is related to defect in the homologous recombination. Yes, ataxia telangiectasia like disorder, Ata tail is present at home. Nis-Megan break syndrome, Nis means apni, Megan Maggie, apni Maggie khud banao. Yes, Ata tail. Yes, what are you doing nowadays? Yes, I am doing my own Maggie I cook. Yes, Ata tail I know. Nis Maggie, apni Maggie, bloom, jhadu, broom, burner. Yes, tea, I make tea for my wife, burner. Yes, roti. Mood kharab hai, wife ka rot mund. If mood is not okay, I will make roti also. Rot mund Thompson syndrome and breast cancer susceptibility. So all these are good of home. Yes, even breast also. Yes, so homologous recombination, all these things are related to home. Mnemonic. Now, nucleotide excision repair. In this, defects mnemonic first of all. Nucleotide T4 tide, T4 tri 0 calorie coke, tri for trichothiodystrophy, 0 calorie xeroderma pigmentosa, coke cocaine syndrome. Question is asked on xeroderma pigmentosa more frequently. Due to ultraviolet rays, there is production of thymidine dimer. This thymidine dimer is corrected by nucleotide excision repair. If this nucleotide excision repair is defective, this lead to xeroderma pigmentosa. So thymidine dimer due to ultraviolet rays, if this repair mechanism defective, this is normally corrected by nucleotide excision repair. If this is defective, xeroderma pigmentosa, in these patients there is a blister on sun exposed area, skin rashes would be there, susceptibility to skin cancer. This clinical scenario they will give and they will ask, yes, which disorder is this? Xeroderma pigmentosa. Which DNA repair defect mechanism is there? Nucleotide excision repair. Mnemonic try 0 calorie coke. So I have discussed this is double stranded DNA break repair mechanism. These are single stranded break repair mechanism. One is nucleotide excision repair, we have discussed. Second is base excision repair. If this is defective, then it is MUTYH associated polyp. I remember say muthi. Your basic power is in your muthi, don't take it otherwise. Yes, basic power in muthi. Mismatch repair, that lead to, yes, defect lead to hereditary non-polyposis colorectal cancer or Lynch syndrome. Yes, your miss will take lunch for you. Yes, will bring lunch for you. Make any mnemonic. Just remember simple single liner questions, sometime clinical scenario, just like xeroderma pigmentosa are asked. Now we start transcription. DNA to RNA synthesis is called transcription, or RNA synthesis is called transcription. We know central dogma. DNA is converted to DNA, that is DNA replication. DNA to RNA synthesis is called transcription. Transcription. RNA to protein synthesis is called translation. Translation. So protein synthesis is translation, RNA synthesis is transcription, and we found that this transcription and translation combinedly gene expression means gene or DNA if want to do function, it will synthesize RNA. RNA will synthesize protein and protein will do the function. DNA can't do the function. Whenever DNA want to do function, it synthesize RNA, RNA synthesize protein and protein do the function. So we are discussing DNA to RNA synthesis. So this is double-stranded DNA. We want to synthesize messenger RNA from this. First of all, these two strands should be separated. And out of these two strands, one strand is coding strand. Suppose this is coding strand, and lower strand is suppose template strand. Coding strand is master. Give order to template strand, go and make messenger RNA just like me. So in this double-stranded DNA, suppose this is coding strand, this is template strand. Now we want to do transcription, means RNA synthesis. These two strands will be separated. These two strands are separated. Now synthesis will occur on template strand. This is coding. Master will not work. This template strand will work. On this template strand, this RNA polymerase, this bulb is just like RNA polymerase. This RNA polymerase don't require primer. DNA polymerase require primer. RNA polymerase don't require primer. Question. RNA polymerase don't do proofreading. Now this RNA polymerase synthesize RNA by this template strand. Now you know the base pairing rule. T will bind with A will bind with T. No, this is RNA. Yes, you know that DNA is having T, RNA is having U. So base pairing rule, A will pair with U, C will pair with G, A will pair with U, G will pair with C, T will pair with A. So in this way, this RNA polymerase will synthesize messenger RNA, and when we saw this messenger RNA, we found that nucleotide sequence was just like coding strand. Yes, I told you, coding strand gave order to template strand, go and make messenger RNA just like me. So this messenger RNA should be just like coding strand, except T is replaced by U. So you can see A, here is A, T is replaced by U, here is G, here is G, T is replaced by U, here is C, here is C, A. So messenger RNA is just like coding strand, except T is replaced by U. So if question is asked and they give coding strand, the base sequence of the coding strand of DNA used for transcription has base sequence GTG. What is the base sequence of RNA product? This is the simplest question. If they have given this is the base sequence GTG, this is coding strand, and you know messenger RNA would be just like coding strand, except T will be replaced by U, GUG. Yes, 5, 3, 8. So we found RNA product is exactly just like coding strand, except T replaced by U. But in the same question, if they change coding word with this is template strand. So if they give template strand, now it becomes difficult. You have to write this is template. Yes, template. Now synthesis will occur on this template. 5. G will pair with C, T will pair with A, G will pair with C, 3 and. So CAC, this would be the nucleotide sequence of messenger RNA. So if they have given template word, the answer would change to CAC. So we have discussed transcription means DNA, from DNA, RNA has been synthesized. But this RNA will not directly enter in protein synthesis. First of all, this RNA will be modified. So after transcription and before protein synthesis, whatever modification will occur in this RNA, that is called post-transcriptional modification of messenger RNA. So we found that in our genome, yes, human genome is very, very large. There is only 1 to 1.5%. If question is asked, how much percent? 1% is the best answer, is exon. Exons are expressed, means exon take part in protein synthesis. So we found that in our genome, only 1% is exon which take part in protein synthesis. All other, yes, introns are there. Introns don't take part in protein synthesis, so these should be removed. First point. Question is asked that mitochondrial DNA, I told you, in mitochondrial DNA, no introns. In prokaryotes, no introns. That is the reason that in mitochondrial DNA and prokaryotes, mutation rate is very, very high. So we found that if mutation occur at this intron, a question is asked, they will give you image that a mutation occur at this point, G is replaced by C, mutation is there. What will be the effect on a protein? You know that introns are not going to take part in protein synthesis, only exons are going to take part in protein synthesis. These introns are removed by splicing. Okay, so we found that introns don't take part in protein synthesis. If mutation occur at intron, there would be no effect on protein because introns don't take part in protein synthesis and removed by splicing. So these introns will be removed. Other than this removal of intron, there are few modifications. Those are called post-transcriptional modification of messenger RNA or RNA processing. First modification means at 5' we will add cap at 5'. At 3' we will add poly A tail. At 3' we add tail. And these introns are removed. Removal of introns means exons are joined, introns are removed. And this is called splicing. So post-transcriptional modification, first is capping, means at 5' we attach 7-methylguanosine. This is important. 7-methylguanosine means methylation is also part of post-transcriptional modification. 7-methylguanosine. This is 7-methylguanosine. Attach at which end? 5' and this is called capping. Question is asked that transport RNA and ribosomal RNA don't require capping. Second modification, poly A tail at 3'. This poly A tail is AAAA 40 to 200 times at 3'. Question, poly A tail is not present in messenger RNA of histone. Third modification is splicing. Splicing means removal of intron and joining of exon. So these introns are removed and exons are joined. This is splicing and this is done by small nuclear RNA. These are ribozyme. You know that RNA having enzymatic activity are called ribozyme. So splicing is done by small nuclear RNA. This small nuclear RNA is having enzymatic activity. So this is ribozyme. So these are post-transcriptional modification. And now the fifth post-transcriptional modification is RNA editing. We know that in our body, in liver, VLDL is synthesized and VLDL contain ApoB100 protein. In intestine, chylomicrons are synthesized and chylomicron contain ApoB48 protein. But we found that in our body, there is only one gene, that is ApoB100 gene. This ApoB100 gene can synthesize both ApoB100 and ApoB48. Question, Previously, we used to say one gene, one protein theory, one gene give rise to one messenger RNA. This one messenger RNA synthesize one protein. But now we started saying no, one gene can give rise to more than one protein. One gene, one protein theory has been discarded. Example is ApoB100 gene. This ApoB100 gene synthesize messenger RNA. This is messenger RNA. In this messenger RNA, capping would be there, poly A tail would be there, splicing would be there. Ultimately, this is mature messenger RNA. Now it can synthesize protein. And we found that in this messenger RNA, there is a codon CAA and there is a codon UAA. UAA is stop codon. Stop codon. You all know 3 stop codon: UAA, UAG, UGA. You are away, you are gone, you go away. These 3 are stop codon. So we found that in this messenger RNA, at this position, stop codon is there. And this messenger RNA in liver will take part in protein synthesis. When this messenger RNA will take part in protein synthesis will continue and will stop at this point when stop codon comes. You know, when stop codon comes, protein synthesis stops. So in liver, we required ApoB100 protein, and this is synthesized from this ApoB100 gene by this ApoB100 messenger RNA, and this is ApoB100 protein. But in intestine, we require ApoB48. So in intestine, the same messenger RNA is there. Same stop codon UAA is there. Same CAA is there. In intestine, there is an enzyme. The name of enzyme is cytosine deaminase. And we have discussed that cytosine and uracil, the only difference is of amino group. If I remove amino group from cytosine, it becomes uracil by enzyme cytosine deaminase. So when cytosine deaminase will act on this messenger RNA in intestine, this cytosine will be converted to uracil and UAA. I just told you UAA is stop codon. So normally this messenger RNA take part in protein synthesis in liver, and yes, protein synthesis stops at this point. But due to this modification, cytosine to uracil, now stop codon appeared early. When this messenger RNA will take part in protein synthesis will stop at this point because stop codon comes earlier by this conversion of cytosine to uracil. And this step is called RNA editing. And the resulting protein would be very small, and we found that the molecular weight of this protein was 48% of ApoB100. So molecular weight of this protein was 48% of ApoB100. That's why it was called ApoB48. So I can say ApoB48 and ApoB100 are synthesized from same messenger RNA. Yes, same messenger RNA. The only difference is this cytosine deaminase RNA editing. So ApoB48 are synthesized from same messenger RNA as ApoB100 by RNA editing. Now question. In the liver, ApoB gene is completely translated to ApoB100. Yes, it is completely translated to ApoB100. In intestine, it is translated to ApoB48. Yes, it is translated to smaller protein ApoB48. Which of the following mechanism explain this? Answer is RNA editing. Now we start operon model. Operon model was given by Jacob and Monod. We are discussing lac operon. This is E. coli. We found that E. coli utilize glucose in normal situation. But in adverse situation, it can also utilize lactose. So normally E. coli utilize glucose, but in adverse condition, when glucose is not present, it can utilize lactose. Gene for lactose utilization are kept in inhibited state. When glucose is there, there is no requirement of lactose utilization. So at that time, gene for lactose utilization are kept in inhibited state by repressor tetramer. Repressor tetramer is attached to operator region. Operator region. What is repressor tetramer? This is E. coli. I have already told you that this is E. coli. In E. coli, there is a gene LAC-I. I means inhibitor gene. This LAC-I gene synthesizes repressor tetramer. This repressor tetramer is attached to operator region. So question is asked, repressor tetramer attached to which region? Operator region. And a question is asked that RNA polymerase is attached to which region? Promoter region. This is a common question. Not only related to lac operon, normally question is asked that RNA polymerase is attached to promoter region. And we found that repressor tetramer is attached to operator region. And I will discuss this in detail. For the time being, cap cyclic AMP complex bind to cap site. Cap site. So these three statements I have told you. Now again come to this is lac operon in which genes for lactose utilization are arranged in linear sequence. And I told you LAC-I gene produce repressor tetramer. Repressor tetramer, each unit is looking like O. O means operator region. They are attached with operator region. This RNA polymerase is attached with promoter region. So this repressor tetramer will repress this RNA polymerase. It will say RNA polymerase, don't synthesize messenger RNA. We don't require lactose utilization. So normally lac operon is kept in inhibited state. Now adverse condition came. Means glucose decreases in environment. When glucose decreases in environment, cyclic AMP increases in E. coli. You can easily remember in human being, when our glucose decreases, glucagon increases, and glucagon increase cyclic AMP. The same way, yes, E. coli are our ancestor. So in E. coli, when glucose decreases, cyclic AMP increases. This cyclic AMP bind with cap. Cap is catabolic activator protein. Catabolic activator protein. It bind with cap to form cap cyclic AMP complex, which attached to cap site. I told you cap cyclic AMP complex bind to cap site. Now, I give you a situation. RNA polymerase is attached to promoter region. RNA polymerase is attached to this promoter region. Repressor tetramer, who is close friend of this RNA polymerase, attached to operator region. Now cap cyclic AMP complex came and it bind to cap site. This cap cyclic AMP activator, catabolic activator, will activate this RNA polymerase and will say go and synthesize messenger RNA. Repressor tetramer would say no, don't synthesize messenger RNA because it is repressor. Suppose you are RNA polymerase and your girlfriend is cap cyclic AMP complex. Your best friend is repressor tetramer. Whom you will follow? Your girlfriend is saying synthesize. Yes, your best friend is saying don't synthesize. Whom you will follow? I know. You will follow your girlfriend. Yes, this RNA polymerase will follow this cap cyclic AMP complex. Why? Because cap cyclic AMP complex indicate that in environment glucose is not there. Either utilize lactose, otherwise E. coli will die. So mnemonic, conceptual mnemonic, cap is more important than repressor tetramer. Means on lac operon, if cap cyclic AMP and repressor tetramer both are attached, then transcription will occur or not. Yes, transcription will occur because cap is more important. Now, this cap cyclic AMP activate RNA polymerase. RNA polymerase synthesize messenger RNA. Messenger RNA synthesize beta-galactosidase, and beta-galactosidase, with the help of beta-galactosidase, E. coli utilize lactose. So this is lac operon. Concept is final concept that normally E. coli utilize glucose At that time, lac operon is kept in inhibited state. But when adverse condition would come, glucose is not there in environment, then this lac operon will be activated, and lac operon will synthesize this beta galactosidase. With the help of beta galactosidase, E. coli will utilize lactose and will survive. But in normal situation, this beta galactosidase is not required, so lac operon is kept in inhibited state. Now question is asked, they will give you situation. If glucose is present, lactose is present. If glucose is there, E. coli don't require lactose utilization, so this lac operon will be kept in inhibited state. Okay. If glucose is present, lactose is absent. If lactose is not there, lac operon definitely will not work. It will remain off. If glucose decreases, is absent, and lactose is present, now E. coli will utilize lactose with the help of lac operon, so lac operon will be on. So now you can simply remember concept that in our body, just like E. coli, many genes are kept in inhibited state. When our body requires those genes, these genes are activated. Otherwise, those genes are kept in inhibited state, and these genes are coming from generation to generation because nature knows that in adverse situation these genes may be required, but when they are not required, they should be kept in inhibited state. Just like E. coli keep lac operon in inhibited state in normal situation, when adverse condition comes, this lac operon is activated. Now we come to nucleosome. DNA is having negative charge. Histone is basic protein having positive charge, and we found basic protein means basic amino acid. Those are arginine and lysine. Arginine is having maximum positive charge. Lysine is intermediate positive charge. Histidine, least positive charge. So if you want to make basic protein, definitely you will take those amino acids which are more positive charge, more basic. So you will take arginine and lysine. So nature decided that histone protein should have arginine and lysine. So don't get confused by the name. Sometimes question is asked and they give histone protein is having histidine. No. Histone, histidine, this is not matching, okay, rhyming, okay. Histone protein is basic protein. It is having arginine and lysine. DNA is having negative charge due to opposite charge. Suppose this is pen is histone having positive charge. This is double-stranded DNA, negative charge. Due to opposite charge, they are tightly attached. And we found that histone are of 5 types: H1, H2A, H2B, H3, H4. So we found H2A, H2B, H3, H4. They make octamer. They make octamer, means 2 subunit of each make octamer, and on this octamer DNA is attached. I told you histone is having positive charge, DNA is negative charge. It is tightly attached. That is nucleosome. That is nucleosome. H1 remains outside this nucleosome. This is called linker histone. So H1 is not involved in this scenario. So nucleosome is made up of histone octamer and DNA. Yes, DNA packing is done by which of the following? Answer is histone. Now this is octamer of core histone. Yes, and this is core DNA. This is linker DNA, linker histone. Now concept of epigenetics. I have given already idea, but I will first explain few concepts, then I will define epigenetics. Recently asked question and topic. Euchromatin and heterochromatin. If I take a cell, resting state, cell is not dividing, cell is an interphase. I saw the nucleus. I found that there was no chromosome. In resting state, in the cell, the genetic material is present in the form of chromatin. Chromatin may be euchromatin or heterochromatin. Lightly stained euchromatin. Darkly stained heterochromatin. Or we found that euchromatin is relaxed, heterochromatin is condensed. Euchromatin is transcriptionally active, heterochromatin is transcriptionally inactive. Means suppose this is histone protein, pan is histone protein having positive charge, my double-stranded DNA, these fingers are double-stranded DNA having negative charge. Due to opposite charge, they are tightly attached. Tightly attached means condensed. Condensed means heterochromatin. Now, if you want to synthesize messenger RNA from this DNA, you can't synthesize. I told you when transcription occurs, these two strands are separated. One strand acts as template and then synthesis occurs. So if DNA is tightly attached to histone, you can't synthesize messenger RNA on it. That's why we found that heterochromatin is condensed. Heterochromatin is condensed and this is transcriptionally inactive, means DNA negatively charged, histone positive charge, tightly attached. Transcriptionally inactive means this DNA will not take part in RNA synthesis. But I can convert this heterochromatin to euchromatin. How? If I take one acetate group and attach to histone, positive charge of histone will be lost and DNA will relax. So suppose this is histone having positive charge and you attach acetate CH3CO minus, that negative charge will neutralize this positive charge. When histone is neutralized, DNA having negative charge will not attach to histone because histone is now neutral. And this DNA will relax. Relaxed DNA means euchromatin. Yes, euchromatin is transcriptionally active and when it is separated, it can synthesize RNA. So we converted heterochromatin to euchromatin by acetylation of histone. If I do acetylation of histone, heterochromatin will be converted to euchromatin. This was a question which is true about histone acetylation. Increase euchromatin formation. Now, if I want to convert it back, means if I do deacetylation of histone, deacetylation of histone, means if I remove these acetyl group from histone, then again histone will have positive charge, DNA negative charge, it will tightly bind. Again, this is heterochromatin, condensed form, transcriptionally inactive. Means we can convert heterochromatin to euchromatin, euchromatin to heterochromatin by acetylation, deacetylation process. This is a part of epigenetics. Point it out. This is a part of epigenetics. What is the definition? Discuss. For the time being, part of epigenetics. We found that we can regulate this RNA synthesis, means transcription, by modification at DNA level. At this time, I told you modification of histone. Modification of histone. Now I will discuss modification of DNA. In DNA, this is promoter region. On this promoter region, there is CpG or CG rich area, cytosine rich area. On this cytosine, if I attach methyl group, methylation of cytosine, then on this promoter region, if I do methylation of cytosine, now this gene will not be transcribed, means methylation of cytosine inhibit transcription. Okay. So you can regulate gene expression by modification of DNA. I told you methylation of DNA, decrease transcription. By conversion of euchromatin to heterochromatin, heterochromatin to euchromatin, by modification of histone. So now I am telling you epigenetics. Epigenetics is reversible. Yes, I told you. Euchromatin to hetero, hetero to euchromatin. Heritable. This is coming from generation to generation. Yes, few genes are kept in inhibited state in our body from generation to generation. Why? When adverse condition would come, these genes will be activated. Otherwise, they are kept in inhibited state. Now, this is reversible, heritable chemical modification of DNA or histone without change in nucleotide sequence. I have not changed any nucleotide sequence. I have just done methylation of DNA, acetylation and deacetylation of histone. No change in nucleotide sequence. This is epigenetics. This question was asked. Which of the following can alter gene expression by methylation and acetylation but no change in gene sequence, nucleotide sequence? Answer is epigenetics. Now, I tell you the significance of this concept. I told you that in our body, many genes are kept in inhibited state. Why they are kept in inhibited state? If adverse condition comes, this gene is activated. I told you that you can convert heterochromatin to euchromatin by acetylation of histone. So, suppose this gene was inhibited state. Adverse condition came and we know we can destroy this adverse condition if I express this gene. So this gene will be activated by conversion it to euchromatin. It will synthesize messenger RNA. Messenger RNA will go to cytosol. It will make a protein and protein will destroy this toxic stimulus. Clear? If this adverse condition is gone, then this gene is again inhibited. How it is inhibited? Again, we will do deacetylation of histone. Yes, and again it will be converted to heterochromatin condensed form and again it will be inhibited. So now you must know the concept. Why? Because they are asking clinical scenario and difficult conceptual question. So in our body, few genes are kept in inhibited state, which can be regulated by acetylation, deacetylation of histone and methylation of DNA. This is epigenetic phenomena. Now, epigenetics and genomic imprinting. Genomic imprinting is epigenetic phenomena. Imprinting means inhibited. This genomic imprinting is a method of gene regulation. Two alleles are present for any gene, one from each parent. We know that in our body, each and every gene is having two alleles and most of cases, both alleles are expressed. But in less than 1% of case, only one allele is expressed, other is imprinted. Imprinted means kept in inhibited state. How you can keep a gene in inhibited state? Yes, I told you, if I do methylation of cytosine, this gene will be inhibited. Yes, this gene is inhibited, to inhibit transcription. So we found that in imprinting, most common method of imprinting is methylation. So what is paternal imprinting? Two alleles are there for a gene. If allele from father is imprinted, inhibited, means if you do methylation of father allele, then it would be inhibited and allele from mother is working. So we will say paternal imprinting. Maternal imprinting means allele from mother is imprinted, inhibited state, but allele from father is working. So this is called maternal imprinting. I take this chromosome 15. On this chromosome 15, this gene, two alleles are there. This maternal allele is imprinted by methylation. Yes. So this maternal allele is not working and paternal is working. Father's allele is working. So in a Prader-Willi syndrome, before Prader-Willi syndrome, in normal situation, chromosome 15, maternal allele is imprinted, inhibited. I give you example. And only paternal allele is working. This paternal allele is working. In Prader-Willi syndrome, paternal allele is also deleted. If I delete this, means maternal is already inhibited and paternal who was working is deleted. Now no working gene. Yes, resulting both copies, father and mother of gene not working, and this will lead to Prader-Willi syndrome. What is Angelman syndrome? Under normal situation at chromosomal 15, this chromosome 15, yes, paternal, this is paternal allele. Another gene I have taken, this paternal allele is imprinted. Now mother is working, mother allele is working. If mother allele is deleted, now both copies will be lost, means both of them not working. So Chromosome 15, yes, paternal allele is imprinted or inhibited and only maternal allele is working in this case. In Angelman syndrome, maternal copy of allele is deleted, leading to various sign and symptom. Means maternal is deleted, paternal was already imprinted, no working allele, means that will lead to Angelman syndrome. Mnemonic: Prader, villi, P4 Prader, P4 paternal, allele is deleted. So imprinting may you will remember deleted. So P4 paternal, P4 Prader villi deleted. Paternal is deleted. Mothers are angel. So in Angelman syndrome, maternal allele is deleted. This question need PG24. They gave that this is paternal. P4 paternal, paternal allele is deleted. So if paternal allele is deleted, this is Prader-Willi syndrome. Now we come to microRNA. Important concept. Suppose there is a cell. In this cell, this gene is kept in inhibited state, and we know that this gene can destroy a particular toxic stimulus. So in this cell, if this toxic stimulus came, this gene will be activated. Normally, this gene is kept in inhibited state, but when adverse condition comes, this gene is activated. I told you that in our body, many genes are kept in inhibited state. When they are required, they are activated. So this gene is kept in inhibited state. Adverse condition came, this gene is activated and synthesize messenger RNA. Messenger RNA come out of the nucleus and synthesize protein. This protein destroys this toxic stimulus. Now I take situation that this toxic stimulus came. Same situation I am telling you. This toxic stimulus came and this gene is activated. This synthesize messenger RNA. Messenger RNA come out of the nucleus. Now it was about to synthesize protein. Till now protein is not synthesized. Till now protein is not synthesized and this toxic stimulus is gone. This toxic stimulus was transient. This toxic stimulus was transient. When this was transient, this messenger RNA should not synthesize protein because we know that toxic stimulus is gone. So now this messenger RNA is not aware that this toxic stimulus is gone. Now we want to inhibit this messenger RNA. How you will inhibit this messenger RNA from protein synthesis? That is done by microRNA. MicroRNA will come and will bind this messenger RNA and will inhibit protein synthesis. Why it is required? Because this messenger RNA, I am not concerned about this messenger RNA, but once this messenger RNA will produce protein. This protein will disturb the function of cell. So we want that when toxic stimulus is gone, this messenger RNA should not synthesize protein, and that can be done by microRNA. So microRNA inhibit this messenger RNA. So now understand the concept of microRNA. Suppose this gene is activated, synthesize messenger RNA. Now we don't want protein from this messenger RNA. How we will inhibit this messenger RNA from protein synthesis? We found that in messenger RNA, there is untranslated region at 5 end, there is untranslated region at 3 end. Normally we don't discuss this part, but we found that in a messenger RNA at 3 end, there is untranslated region. And microRNA is very small. Name itself tells you micro. It is 21 to 23 nucleotide long. This is microRNA. This microRNA will come and bind to this 3 untranslated region of messenger RNA and will inhibit this protein synthesis. Okay. So first question, microRNA bind to 3 untranslated region. First question. MicroRNA causes gene knockdown. So this phenomena in which this messenger RNA is not synthesizing protein, or this messenger RNA is inhibited from protein synthesis, this is called gene knockdown. Now make mnemonic. Mnemonic is that microRNA M. If I rotate once M, it will 3. So microRNA bind to 3 untranslated region. First question. If I rotate 3 once more, W, gene knock down. Gene knock down. W, down. So question is asked, mechanism involved in RNA interference or microRNA is gene knock down. Why I have told you this W? Because you will find option knock in, knock out. At that time you will get confused. So when you will remember W is present only in down. It is not present in and out. So microRNA cause gene knock down. And I told you that microRNA, another name is RNA interference, means microRNA is interfering this messenger RNA from protein synthesis. So that's why microRNA is also called RNA interference. Another name of microRNA is RNA-induced silencing complex. Means by this microRNA, this messenger RNA is silenced. Okay. Next name is small interfering RNA. This microRNA is very small and it is interfering the normal process of protein synthesis from messenger RNA. So I can say this is small RNA interfering another RNA, small interfering RNA. Small form is siRNA, small interfering RNA. Another name is silencing RNA, means this microRNA is silencing this messenger RNA. Silencing means it is not allowing this messenger RNA to synthesize protein. So now multiple name and the function is same. Question is asked. So remember, microRNA, small interfering RNA, silencing RNA, siRNA, RNA-induced silencing complex, concept is same. Little bit difference is there in their origin, but concept is same. Question 3, untranslated reason, gene knockdown, recently asked question. Now, in protein synthesis, there are 64 codons. Out of 64 codons, 61 code for 20 amino acid and there are 3 stop codons. 3 stop codons are UAA, you are away. Another name is Okri. UGA, you go away, Opal. You are gone. Yes, UAG, you are gone. That is amber. We found that selenocysteine 21st amino acid, yes, codon is stop codon UGA, yoga of Selena Gomez. Yes, so UGA is stop codon but can be recorded to selenocysteine. UAG, AG, pyro is also used for AG. So UAG can be recorded to pyrolysine. We found that 61 code for 20 amino acid. If I take average 61 divided by 20, on an average 3. So 20 amino acid, 61 codon. If I take average, 1 amino acid on an average is having 3 codon. So I can say for 1 amino acid, there is more than 1 codon and this is called degeneracy of codon. Means more than 1 codon for single amino acid. I take example, UCA code for serine. And we found that UCU again code for serine. Means UCA and UCU, there are 2 codon for single amino acid serine and this is called degeneracy of codon, more than 1 codon for single amino acid. And when we saw that in this serine codon, UC portion is same, the third nucleotide is changed. So a statement is asked that third nucleotide of a codon is not so specific. What I said, third nucleotide of a codon is not so specific, means in serine, third nucleotide is changed and we found that both codon code for same amino acid serine. Means if you change third nucleotide, there are chances that the new codon may code for same amino acid, just like UCA and UCU code for same amino acid. We found that most of the amino acids are not loyal. Means they are having more than 1 codon. But yes, these 2 amino acid, methionine and tryptophan, they are loyal. They are having only 1 girlfriend or boyfriend, that is methionine and tryptophan, only 1 codon, means they don't show degeneracy. all other amino acid, and out of these, serine, leucine, arginine, they are playboy, playgirl, means more than one codon, and they are having six codons. Yes, so serine, leucine, arginine, 6 codons are there, means they show degeneracy. There is a concept of Wobble hypothesis. If I say this is messenger RNA, in this messenger RNA, there are 2 codon, UCA and UCU, both code for serine. So when protein synthesis will take place, at both position, this codon will code for serine. We know that transfer RNA have anticodon. We found there are 31 transport RNA. There are 61 codon which code for 20 amino acid. So if I take average, 61 divided by 31, approximately 2. So we found that approximately 2 codon are represented by only one anticodon. So suppose we take simplified example. If you don't remember the example, it's okay. Just have an idea concept. Serine is having only one anticodon, suppose. That is UGA. And serine is suppose having only 2 codon. That is UCA and UCU. Now when protein synthesis will occur, it would be difficult to synthesize protein if Wobble hypothesis is not there. Animation. Suppose this is messenger RNA. In this messenger RNA, these are 2 codon coding for serine. Now transport RNA, only one anticodon is there, means one transport RNA type is there. This transport RNA having serine will come and will bind with this codon. So anticodon bind with codon, and we found that this is totally matching. Yes, base pairing rule is followed. U bind with A, G pair with C, A pair with U, means base pairing rule is followed. Now UCU again code for serine. Now we have only one anticodon. Now what will happen? Second transport RNA of serine will come, and it will try to bind with this. The confusion would be A will pair with U. This is base pairing rule. G will pair with C, base pairing rule, but U will pair with U. Yes, this is abnormal base pairing, but there is no other option because we are having only one anticodon for serine and 2 codon for serine. So this is required, that abnormal base pairing should be there between this first nucleotide of anticodon and third nucleotide of codon. So this is Wobble hypothesis. Wobble hypothesis says that abnormal base pairing can occur between first nucleotide of anticodon and third nucleotide of codon. U and U base pairing occur. This Wobble hypothesis. Yes, abnormal base pairing can occur between third nucleotide of codon and first nucleotide of anticodon. This is Wobble hypothesis. Third nucleotide of codon is not so specific, and this Wobble hypothesis is required. Why it is required? Because codons are degenerate. Yes, more than one codon for single amino acid. This deep concept was asked. So what was the exact question? We don't know. That's why I took a little bit more time on this, because 2 times question has been asked. So just understand the concept. Now we find different types of mutation. Silent mutation. Silent mutation means codon for one amino acid is replaced by codon for same amino acid. Suppose I take UCA code for serine and mutation occur and UCU again code for serine. Due to this mutation, previously there was serine, even now there is serine. So codon for one amino acid serine is replaced by codon for same amino acid serine. This is silent mutation. If mutation occur and UCA is converted to UAA, UA is stop codon. Stop codon. So if there is production of stop codon by mutation, that is called nonsense mutation. Nonsense mutation means codon for one amino acid, just like in this example serine, is replaced by stop codon. Yes, UAA, stop codon, that is nonsense mutation. Serine mutation occur and it is converted to CCA. CCA code for proline. So we found that codon coding for serine is replaced by different amino acid proline. If one amino acid is replaced by another amino acid, that is called missense mutation. Means codon for one amino acid, that is serine, is replaced by codon for another amino acid, just like proline. This is missense mutation. Missense mutation may be homologous or conservative, or non-homologous or non-conservative. What does it mean? Suppose amino acid, one amino acid is replaced by amino acid with different properties. Example, sickle cell anemia. In sickle cell anemia, in beta globin chain at 6th position, glutamic acid is replaced by valine. Glutamic acid is having negative charge. So glutamic acid is acidic amino acid. It is having negative charge, polar with charge. Valine is non-polar, branch chain amino acid. So if amino acid are changed in such a way that their properties are not matching, different properties, different characteristic, definitely there will be effect on the function of protein, just like in sickle cell anemia. And this is called non-conservative or non-homologous mutation. Now, conservative or homologous mutation means amino acid is replaced by similar amino acid, just like aspartic acid is replaced by glutamic acid. You know, aspartic acid, glutamic acid, both are acidic amino acid having negative charge. So if they are changed, there are chances that protein function would not be affected much. That is conservative mutation. So sometimes question is asked that there is a mutation in which aspartic acid is converted to glutamic acid, there would be least effect on the protein, or by which mutation, by which amino acid change, there would be least effect on a protein function. If conservative mutation is there, then there would be less effect on the function of protein. Another example of homologous mutation is valine is branch chain amino acid, isoleucine is branch chain amino acid, and both are non-polar. If valine is replaced by isoleucine, there are chances that the mutated protein would not have much effect on their function. Now we come to frame shift mutation. Frame shift mutation means alteration of reading frame. If number of bases added or removed not multiple of 3, then it lead to frame shift mutation. What does it mean? Suppose this is normal RNA, mutation occur and deletion occur. Deletion of this occur. In normal, AUG code for methionine, UCA code for serine, UCU code for serine, UUU code for phenylalanine, AUG code for methionine. Now I told you that mutation occur and this U is deleted. When this U is deleted, the complete frame is shifted. Means first amino acid will remain same, but further amino acid will be changed. Means next codon would be, yes, CAU code for histidine, CUU code for leucine, UUA code for leucine. So complete frame is shifted. All amino acids are changed. This is frame shift mutation. Now I take non-frame shift mutation. If number of bases added or removed multiple of 3, yes, if entire codon is added or removed, then it would not be frame shift mutation. Suppose this is AUG and all these 3 nucleotide are removed, codon is removed. Previously, this was serine, phenylalanine, methionine. Now, what would be the change? Only one amino acid will be missing. Otherwise, serine will remain serine, phenylalanine will remain phenylalanine, methionine will remain methionine. No frame shift mutation if 3 nucleotides are cut or added. Now we come to suppressor mutation, recently asked topic. Suppose this is gene or messenger RNA in this first mutation occur, and due to this first mutation, this UCA, which code for serine, is converted to UAA. UAA is stop codon. So if first mutation occur and due to this mutation, this serine is replaced by stop codon, this is a nonsense mutation. And due to this nonsense mutation, protein synthesis will stop at this point. We want to revert the effect of this mutation. So what nature did? Nature knows that this codon is recognized by anticodon. Means when protein synthesis occur, this UCA code for serine, so transport RNA having serine will come. Normal situation, if there is no mutation, normally transport RNA having serine will come and U will bind with A, U will pair with A, G will pair with C, A will pair with U, base pairing rule is followed, and here protein synthesis will occur and serine will be added. But due to this mutation, now it is stop codon. So protein synthesis will stop. So now what we did, yes, what nature did or body did, it did mutation on this transport RNA. Gene of transport RNA mutation occurs, second mutation in transport RNA gene. If second mutation occur and this previously it was G, now I replace this G with U. Second mutation converted this G to U. Now this transfer RNA will have serine, and now this transfer RNA base pairing rule will be followed. U will pair with A. This mutated now anticodon, U will pair with A will pair with U. Means this anticodon which is mutated, means the second mutation which occurred in transport RNA, this anticodon is changed. Now this changed anticodon will recognize this stop codon and this serine will join at this stop codon. So by mutation in transport RNA, second mutation in transport RNA, we reverted the effect of first mutation. So this is suppressor mutation, a second mutation which is done to revert the phenotypic change done by previous mutation. So previous mutation, this serine codon was converted to stop codon. By second mutation, we did mutation in transport RNA. Now the transport RNA anticodon will recognize this stop codon, and previously there was serine here, and now again serine will join at the same point. So phenotypic change would be reverted. This is suppressor mutation, recently asked question in May 24 INICET. Now we come to protein synthesis. In protein synthesis, three steps are there: initiation, elongation, and termination. But before this initiation, there is a phenomena called activation of amino acid or charging of amino acid. We know that there are 20 amino acids which take part in protein synthesis, and there are 31 transport RNA present in our cytosol. So I can say each amino acid is having its own transport RNA, transport or transfer RNA or CAR. So each amino acid has its own CAR. Few who are rich are having more than one CAR. One person is having VIP CAR, that is methionine. Methionine is having VIP CAR because first amino acid in protein synthesis is always and always methionine, and codon for methionine is AUG. AUG code for methionine. So I leave this. Now again come to my point that transport RNA, suppose this is transport RNA for serine, serine amino acid will come and will bind to this transport RNA, and this is done by enzyme amino acyl transport RNA synthetase. Synthetase ATP is used. Synthase, no ATP used. Synthetase ATP is used. In this, ATP is converted to AMP. When ATP is converted to ADP, one high energy phosphate used. When ATP is converted to AMP, two high energy phosphates are used, equivalent to 2 ATP. So activation of amino acid, 2 ATP used. We found that this amino acyl transport RNA synthetase has proofreading. proof reading. This is driver. You can understand a story. This transport RNA is car. This serine is owner of car, and this amino acid transport RNA synthetase is driver. He is taking money, 2 ATP. So now he will keep in mind that this car belongs to serine. So he will allow only serine to bind it. But by mistake, suppose valine came and valine attached to this transport RNA. Means valine take this car. Now this amino acid transport RNA synthetase driver will see, yes, car is off, serine, who is sitting, valine will remove it. And this is called proofreading. But if this driver, amino acid transport RNA synthetase, forgot to correct it, then abnormal protein will be produced where you should join serine, there would be joining of valine. So point it out. If this amino acid transport RNA synthetase do proofreading and fail in proofreading, then abnormal protein will be synthesized. In place of serine, valine will join in protein. So now I solve question. Amino acid bind at 3 end of transport RNA. It bind at 3 end. Amino acid transport RNA synthetase has proofreading. Proofreading question. Activation of amino acid or charging of amino acid require 2 ATP. Fidelity or reliability of protein synthesis depends on amino acid transport RNA synthetase, because this is the only site where proofreading occur. If by mistake this proofreading is failed, means this valine is not removed in protein synthesis, in place of serine, valine will come. Abnormal protein will be synthesized. Now we start initiation. Initiation of protein synthesis. Initiation means formation of initiation complex. Initiation complex is made up of transport RNA having methionine. I told you first amino acid is always and always methionine, and codon for methionine is AUG. So transport RNA having methionine, ribosome and messenger RNA. So you can say this is initiation, formation of initiation complex. This is messenger RNA. This is transport RNA having methionine, and this is ribosome. This is ribosome. So question is asked, initiation codon is AUG. Yes, I told you methionine codon is AUG. Initiation codon is AUG. And first amino acid is always and always methionine in eukaryote and 4-mil methionine in prokaryote. Now we understand initiation by animation. Formation of initiation complex. First of all, ribosome is separated. Now this messenger RNA and this transport RNA is coming, and in this way all these three component, means transport RNA having methionine, ribosome and messenger RNA join to make initiation complex. But the problem is there, we found that in this messenger RNA, AUG code for methionine, this AUG again code for methionine. So when this initiation complex is being synthesized, there would be confusion that when this transport RNA having methionine will come, it will be confused. I should bind to this AUG or I should bind to this AUG. What is the starting point of protein synthesis? That is determined by a sequence that is called Shine-Dalgarno sequence. Shine-Dalgarno sequence. First question, Shine-Dalgarno sequence is UAGG.